So here we go again. Hi, hello everyone. Hi, I'm Oliver, Microbehunter here. And I think everything seems to be rolling quite fine so that we can start another microscopy live stream this week. Yeah, today I'll do again a little bit of microtomy work. Do you say it like that? I'm going to be using a microtome again because I have grown this Ilodia plant, this water plant here, and I would like to cut it up a little bit using my microtome to look at it under the microscope, of course. I have done this already a couple of months ago. Quickly, I tried it out and it looked quite nice. So I hope I'm able to repeat it today. And of course, I want to make again a permanently mounted microscope slide. So that's basically the goal or the task that I have for today. Before I do that, two important things. Of course, I would like to look at the two slides that we made last week. I used different types of glue to put some algae on a microscope slide here. We're going to check how this actually worked out and if it's still fine. So that's basically, I'll start off with that. And of course, I would like to say hello all around the world. And yeah, so that's basically, if you have any comments, please feel free to write it in the live chat. Also, there are already a couple of comments for me. Greetings from Vietnam as always. Don't forget to do a stem cross section as well. It turned out beautiful the first time and last time you did it. That's the whole point, actually. The stem cross section, which I quickly tried, looked very nice here. I hope I'm able to repeat that, but not only that, I would like to actually make a permanently mounted slide. But it's already starting to dry up on me here, so I'm quickly going to put it back into the jar here. Okay, I don't want it to dry up completely. And yeah, so, but before I do that, I would like to quickly check how the slides from last week actually turned out. Okay, if you remember last week, for those of you who were here, I tried different types of glues, different types of mounting media to put some algae under the microscope. And what I'm going to, for maybe, I don't know if you've seen it last week or not, and for this reason, very quickly, just a couple of minutes, okay? I would like to, for those of you who have not seen this, repeat this a little bit. I still have the algae sample from last week. I'm going to show you a fresh sample, and then we're going to compare it with the permanently mounted slides that I made, simply to kind of, yeah, to check if there's any loss of quality. Okay, it's a little bit the idea. What I've got here now is the sample from last week. Remember, I centrifuged it and I kept it in here. Yeah, so this is a 1 week old concentrated algae. And let's see how this looks like. So it's again a very short repetition of last week, simply to check how the specimen is doing, so that we can actually then compare it a little bit better. As always, I will be always watching the chat with one of my eyes. So feel free to ask questions and yeah, to comment. Yeah. So here we go. Yeah, and it goes under the microscope. And usually, yeah, fresh samples are always best, but maybe this will also be fine. Okay, here we go. I have to turn on the camera. I have to turn on the microscope. I have to focus, of course. And of course, I'm going to go up with the magnification here a little bit. Okay. Yeah, and you see that the sample still looks fine. Over here, okay. The sample still looks, let's increase the contrast a little bit here. The sample still looks fine, obviously. And fresh samples usually always, yeah, the best, obviously, to observe, because sometimes you see some movement. I've done this last week quite a bit, but now I would like to simply have a look now at the two slides that we prepared, and let's check a little bit if the specimen actually, yeah, is kind of degraded or not. Okay, so this is a little bit the thing. So let's put it in here again. This is now for those of you just joined in. I'm just repeating very quickly last week's experiment, and I would like to check. So this one over here, I have to refocus, of course, and I have to find it. Let's see where it is. Here we go. Okay. Let's go to a place where the, yeah, let's go a little bit lower with the magnification here. Ah, yeah, and these are basically, what we see already, something quite interesting. There's a huge bubble here. And these are now the algae, which I made using this glue over here. This glue. You don't see it again because it's of my green screen. Yeah, I quickly show it to you here on the desk. Here, that's the glue I used. It's a water-based glue, and this is the result. Let's remove the DIC. So this is basically a regular bright field, and I think it's okay. I mean, yeah. Of course, the specimen will always degrade a little bit with time, and it will lose color. But for experimental purposes, it works. Yeah, this algae over here that you see here, that's quite nice. It's called Pediastrum. It's a nice colony-shaped algae. And so the next one is I have to check now. I have to check on this one. The next one is it's Elmer's PVA glue. It's also a water-based glue. Yeah. Looks okay. You see the cyanobacteria, those long filamentous, blue-green algae as they used to be called. They're a little bit, I don't know, crooked and not quite as straight anymore. Yeah, so that is the other one, the Elmer's PVA glue. And the last one, this one over here, I have to check what is that. This seems to be, glycerin gelatin. Yes. This glycerin gelatin, it's the same mounting media that I'll be using today. It looks like this. Yeah. It's glycerin gelatin. It's a mounting medium that does not turn completely solid. It does retain a little bit of moisture as well. And sometimes you are able to see that some structures, like the one, the circular thing in the middle, I don't know what it is, but some of them will actually turn. In this case, they will turn a little bit reddish because there is a dye mixed in, a stain mixed in into the mounting medium. Yeah. So that is basically, you see that all three of them, are water-based. Yeah. And I've got another one over here with glycerin gelatin. Okay. Exactly, yeah. And this one over here, it's another one with glycerin gelatin. I have to find it again. Let's go down with the magnification a bit. Here we go. Yeah, the density is a little bit lower here. Yeah, so you see those, at least, they are able to store for 1 week. But again, as expected, the quality is not quite as nice as the fresh specimen, obviously. Yeah, but that was to be expected. So that's the fresh one that we've got here. Yeah. Of course, there's a little bit of movement going on, which also makes it a little bit more interesting. And you see that the green color is a little bit stronger here as well. Yeah. So yeah, just basically wanted to recap a little bit from last week. Okay. Yeah. Let me check. Yeah, some of you I see have actually also ordered it, the glycerin gelatin. It's a little bit difficult to use mounting medium, unfortunately. Yeah. So I just quickly read some of the comments. Yeah. I tried to make my own glycerin gelatin, but you cannot and should not use the gelatin that's used in food. For example, you can of course buy gelatin. You can of course buy gelatin in also for making cake and so on. But what I read is that the pH is not quite correct, right? It's so you should actually ideally use some pure gelatin powder, which is laboratory grade, and then you also have to add some kind of a disinfectant in here as well. Yeah. So yeah, this was a little bit of a recap of last week's how do you say observations. And what I would like to do now, before I actually start trying to cut up the the water plant, which looked kind of nice. I did discover something here, which I also want to show you. I mean, I got this a couple of months ago. This water plant, and I had to exchange the water frequently because if you don't do that, it actually starts to degrade a little bit and starts to decompose. But then all of a sudden, look what I found here. I don't know if you're able to see this. Look at this. This was not present a couple of weeks before. Look at this. These are filamentous algae. This is all of this stuff has grown in the jar. So I have to probably remove it, otherwise it's going to overgrow it. I don't want to throw it away either. But I'm just going to take a small sample here. Yeah, and we're just also going to put it under the microscope. These are filamentous algae, and they have rapidly grown over the course of a couple of weeks, which is yeah, also to be expected a little bit. Let me add. I've got here. Here, let's go with this one over here. Add a little bit of water here, of course, and I'm going to now use my huge cover glasses. And I usually, before I dump things out and throw them away, I always put them a little bit under the microscope to see what we're able to find. And here we go. That's a huge cover glass. Look at this, huge one. It's of course way too thick. I'm going to press it down. Let's have a look at this here very quickly. So here we go. Go down with the magnification. So it's way too bright. I got some other plants here as well. So these are clearly, filamentous algae. I got something of the Illudia as well. And a little bit of movement. So there seem to be also other organisms present. Yeah, but you see that's a little bit. I was actually hoping to find a specific type of filamentous algae called Spirogyra. Unfortunately, they have a very beautiful spiral-shaped chloroplast, but this one as well looks quite nice. It's okay. Yeah. So that's a little bit as expected. Yeah. And now I'm going to simply take it and I'm going to throw it out. Yeah. Yeah. And because otherwise they're going to grow so quickly that sooner or later it's going to probably choke all of the remaining water plant here. Yeah. Okay. I always see that you just drop the cover glass on the slide, but I read and see everywhere that you should not do that. Does the technique not matter? Let's put it this way. It's a good point. I'm sometimes very fast and sloppy about certain things, and they always tell you, okay, if we already talk about adding cover glasses, so this is one of the comments here. How should you put on a cover glass on the microscope slide? Usually, let me remove this one over here. What they tell you to do is you take a small sample of material. You know what? Let's do that with one of the leaves here. Yeah. So let's take, I don't know, this is a very small one, right? Let's put it on here. Yeah, let's add a little bit of water. Yeah, and what you have seen me do is I take a cover glass, right? And I basically over here, look. And I basically do the following. I basically just drop it on here. What I do is the following, and you might not be able to see that. I do hold it at a slight angle, right, downwards, and then I drop it. What they always say is you should lower it carefully, because if you drop it completely flat, you might catch some air bubbles. And by holding it like this, kind of a little bit, and dropping it like this, then you're kind of pushing out the air. I've found it doesn't really matter. Normally what they do is also, I'm a teacher, so when we instruct students to do this, we always tell them to kind of carefully, let me take it off again, carefully lower the cover glass at an angle, right? You're supposed to hold it like this, and you're supposed to carefully lower it and then drop it on here. I think as long as the specimen is sufficiently thin, it's going to be fine. Yeah. And another thing that you don't want is you certainly don't want to have any water on top. I consider that to be a much bigger problem, actually. Yeah, because then you cannot really go up with a magnification. Yeah. So what I'm going to do now is we're going to quickly look at this here, and then I want to make a couple of cross sections. This you have also seen already before. It's also one of my more favorite specimens. Is of course are the chloroplasts of this water plant of Ilodia, or Ilodia densa, or Egeria densa, as it's called. And what you see, it's a nice specimen because the leaf is sufficiently thin that you can make a whole mount without cutting it. And let's see here, maybe at the side somewhere. Yeah. We find a space where the density is not all too high. It doesn't matter. We go up here. And what I have done already before is I've made some beautiful time-lapse images where the chloroplasts are moving due to cytoplasmic streaming. This basically is a movement of the cytoplasm inside the cells, and they're pulling the chloroplasts along. It looks quite nice on the time-lapse. Over here, we don't see it quite as well. I think the density is quite high of those chloroplasts, but if you look very carefully and patiently, you're going to see that they actually do move. Yeah. And concerning bubbles, another thing is sometimes you try to avoid bubbles, and then you have some algae or some water plant under the microscope slide, and then all of a sudden you see those bubbles again appearing, and they actually start to grow because this is actually oxygen that is being produced by the plant. Yeah? So you try to avoid bubbles, but then actually you get them back again by photosynthesis. Yeah. Yeah. So this is simply something here. I'm going to quickly answer some of the questions, and then I do want to cut up a little bit of the plant here. What can I use to fix objects to the slide without heat so that the water does not move the position when placing the cover glass? That's difficult. I want to quickly explain this. The problem is the following: when you have a specimen on a slide, and which is small, and then you put the cover glass on top, exactly at the angle which I showed you, right? Then there is sometimes the problem, very often the problem, that as the cover glass is lowered, it pushes the specimen to the side. And then you have got the specimen right at the edge. The question is, what can I now do to prevent this from happening? What I usually do is I lower I put the cover glass, I put the specimen not directly in the center, but at the edge of the cover glass, so that when it is pushed to the side, that it will end up towards the center. So it's a little bit of trial and error. Sometimes I've also tried to use a little bit of mounting medium if I make a permanently mounted slide, to kind of glue the specimen a little bit to the slide before I add the remaining mounting medium. This is also something that you can try, and it depends a lot on the specimen itself. But I get the problem. I've experienced this myself. You put something on the slide, you put the cover glass on top, and then everything's completely pushed to the side. Yeah. Could Ilodia regenerate from a single leaf? No, it cannot. But I will show you what it will do. It can regenerate, but not from a single leaf, to my knowledge. Let me quickly show you. As a matter of fact, this is a very nice to use water plant because it does grow very quickly. And because it grows so quickly, it also absorbs a lot of nutrients from the water. And this leaves less nutrients for algae to grow, right? So what you have here is, let's take this one here out. Let's put it over here. Yeah. And then I want to start cutting it up a little bit. It will actually start growing, and you see over here at the side, sometimes, well, not here really, sometimes you actually see that it starts to branch off, and there is going to be a side growth. And I don't see one over here now. Let's try another one. This one is quite small. Maybe I'm going to use this one for cutting. But it will actually start to branch off. However, what I also found that's really important, this one here that I have, it does not look very healthy anymore. And I think one of the reasons is because there are already so many other algae growing, and somehow, look, this whole mat of algae here, it starts to choke it. So what I have to do, probably, I'm going to remove all of this here first, and untangle it. But if you observe, check it, then sometimes you will see that it will start to branch off. Yeah, and so let's take this away. Yeah. If there are too many of these, then they will, of course, not only consume a lot of nutrients, but also light, especially. And then, because you see it entangles everything so much, the plants cannot really properly grow. Okay. Look at this. Wow. Yeah, again, this was not present a few weeks ago. Yeah. It almost feels a little bit of a pity throwing it away. I'm not going to throw it away. I'm just going to put it on here. Yeah. Now it looks a little bit better. So what I'm going to do now is the following. This is now going to be a little bit of an interesting challenge, because I want to try to cut this, and I need to find, yeah, I'm going to simply take part of the stem here. Okay. Just going to maybe use my fingernail to take it off. Let's put this away. You know what? I'm gonna get myself another Petri dish. Here we go. I'm going to put this here away. So let's do it like that. I've got some water here. It'll be okay. And now the big challenge is how am I supposed to put this into the microtome, right? And I will use some. Everything's wet here already. So, number one, my microtome is here, and this little piece I misplaced. Where did I put it? Here it is. Okay. So what I now need to do is I need to put this into the microtome. I don't know. I'm just going to explain it again. As 3D printed, this is a specimen holder. The two holes in here are actually for the tweezers so that I can take it out more easily. Okay. So yeah, and the question is, are we mainly doing stem cross sections, or is there something else? Well, we're gonna see. It depends a little bit on what you want to observe. My plan is actually to mount this in glycerin gelatin because I've never mounted a stem cross section before. That's the thing. Yeah, I've only observed it, but not done a stem cross section as such. So what I'm going to do now is I'm going to remove now the leaves here. Okay. And yes, another thing that I did want to do, but this only takes maybe 5 minutes. I do want to start an enrichment culture of paramecia. I'm going to show you how to do that as well. I've got some grains here, some wheat grains that I would like to use. But this only takes a couple of minutes, and then maybe next week we're going to see if the culture of paramecia has actually grown or not. Yeah. So this is a little bit step one. Okay. So this is step one. So, and I'm going to take this stem now, and that's now basically the thing that where I would like to do a cross section. And for those of you who already mentioned this, somebody already mentioned this a couple of weeks ago when I tried that. It looked kind of pretty and interesting, and I hope I'm able to reproduce that. So I'm going to now use maybe one of those notches here on the side, maybe this one over here. I'm going to put this in here, right? And I'm going to put it into the microtome. Okay. So normally I would clamp this to my table, which I cannot do now, right? And I'm going to now use my sharp razor blade here. I add a little bit of water to prevent the specimen from drying out. And I'm going to simply give it a try now by carefully yeah, cutting it like, you actually see this, it's a little bit so here, now it's, yeah, and it can be cut very easily. I'm going to put it in here just to save it. And now I have to turn it down here a little bit. 1, 2, 3, 4, 5, 6 times. Yeah. No, it was the other direction. So. And here we go. This is this tiny thing that you see here. This is actually already yeah, one that I could try to use, and maybe I'm able to directly put it on a. Maybe I should try to put it directly on the microscope slide. How is that? So I'm going to try to grab it like this and put it directly on here. I turn it again. So no, this wasn't enough. So this is another one. Yeah, the problem is I feel the leaves kind of are not quite, they're a little bit in the way. And this is not a very easy, it's not quite as easy, and I'm a little bit worried that maybe the razor blade has already become a little bit dull, maybe. Let's see. Let me see how this one here is. No, this one is also bad. Okay, we'll try another one. Not happy with this. You know what? Give me a second. I'm gonna try to get myself a fresh razor blade. So I'm back now. Got myself a fresh box of razor blades here. And let's try to get this, unpack them. Here they are. And I need to, this is also something that I 3D printed. It's, yeah, the razor blade holder. Added some 2 screws here to hold it together. And this one is already a little bit older. I've already used it. And I'm going to use now a new one. Yeah, also much less corroded, as you can see here. And this here goes on top. And this here goes here on top. Up. Be patient. So let's give it another try here with a fresh razor blade. I'm going to turn it again. Yes. And yeah, it doesn't really catch it quite well. Okay, here we go. So let's put this down here. So what I'm going to do now is without cover glass or anything, I'm just going to give it a quick check under the microscope to see how it looks like. And yeah. Okay, so this is without a cover glass or anything. Okay. Yep. Okay. At least this one here is definitely a little bit too thick, and because there is not a lot of water here on here and no cover glass for this reason, it does not look quite as nice as one would expect. Yeah. Is this the microtome from Brunel? Yes, it is. I also bought this from Brunel. And I can talk a little bit about this microtome because they actually recommended that I use a carrot as a specimen holder, which I did not do, and that's why I 3D printed this specimen holder in here. But let's try a few more because I would like to do the following. I'm going to take it out now because this is a little bit already not so good. So let's put this in here. I'm going to now put it further down. This is actually a pretty good microtome. I'm quite happy. You can actually see you can use it here to clamp it to a table. And yes, number one. And number two. Yes, now the cuts actually work much nicer. Number three. Number four. Here it is. Number five. This one was a little too thin. Yes, much better. Generally, my recommendation is always trial and error. The more that you have, the more cuts that you make, the higher the chance that a few of them will actually look nice. Yes, okay. Okay, yes. So, I mean, there are plenty of them here. I'm just going to save them now on a slide. Which slide I'm going to use here? Maybe this one here. Here we go. And here are also a few. Here we go. No, there are lots of them now. Yeah. And next one. Okay, this looks quite okay. And just for experimentation, I would like to now make a few cuts that I will now directly observe without making a permanent mount. Okay, here this one. So this half cut. Okay. Okay, lost it again. Let me put a drop on here. Yes, here it is. Yes, here we go. I'll put now a cover glass on top, of course. Yeah. I add a little bit more water here because it's not. Here we go. So this is basically the temporary mount now with cover glass to simply do a little check how it looks like. Here it is. It's actually pretty okay. It's pretty decent. Yeah. Yeah, it's not. It's a little bit cut diagonally, but the idea is now am I able to make a permanently mounted slide here. Yeah. Let me quickly check some of the comments here. I notice seeds. Oh, I don't know what you're referring to the middle edge. I don't know now. I did not check the comments here. It's the microtome. So that is the question now is instead of a carrot, could you put a specimen there and basically fill the whole thing here with paraffin wax? This is a little bit the question. I mean, you put a specimen in here, right? You put a specimen in here, and whatever the specimen is, and you kind of fill it in with wax, and you allow it to cool. My concern is a little bit those edges here that you see on the side. That it could be that maybe the wax is actually going to run into it here. But what you can do is if you get some kind of a mold and then put the paraffin wax, if you're able to make it outside of this microtome and then put the solid block of wax in here, that should work. Huh? But actually putting the molten wax in here is probably not a good idea because it might actually run down here on the edges, right? And then it goes into the mechanics here. You probably don't want that. Yeah? But I know that officially they recommend recommend a carrot, which also works. Yeah, but it is messy. Yeah. So that's the thing. Yeah. So let's do the following. I've now made those here, and I don't know which ones are good and which ones are not good. So it doesn't matter. I'm just going to try it. I'm not here to, this live stream is more a little bit always like a proof of concept, really. It's not about perfection, because if you want to get a really nice specimen, then you really have to take a lot of time and so on. It's a time that I obviously don't have during this 1 hour here, but I hope that at least if you see how it works, you at least get the idea to try it out yourself. So what I now need is the following. Did I plug this in already? I need to again need the warmer. Yeah, it's already plugged in. So let's do this here again. And now I need to warm up a slide, and I'm going to put it's again, you're not able to see it. I'm going to put a little bit some of this mounting medium on here. This is indeed, I have to honestly admit with you, never tried this before. I've not done it with this specimen yet. So in that sense, it is as also for me a little bit, a new thing to experiment around. Maybe a year or two ago, maybe some of you remember, I actually tried to melt the whole jar. It takes a long time, and then there's always the danger of overheating. So I'm not doing that anymore. I found it much more practical simply to take a spatula and remove a small amount of the solid mounting medium directly from the jar. Yeah. Like this. Yeah. Maybe this is already a lot. Huh? This is already very hot. It's already very hot, so I don't want to overheat it. Yeah, and it's already melting. See. So, and the next trial that I have, I'm just going to see if I'm able to put some of this stuff here directly. I don't know which one's good and which one's not good. Directly into the mounting medium. Again, I've not checked the quality of the cross sections here, so some of them might be okay, others might not be so good. And then maybe I'm going to actually also damage some of them as I transfer them. I think that's all possible. But for me, my goal is not really necessarily to make now a very beautiful looking slide, but more to check whether this mounting medium is actually suitable for this type of specimen. It's difficult to pick up. Yeah, because of course there's going to be probably a little bit of damage done as well. Yeah. So, and then a cover glass goes on top. Yeah, you see, you already see a problem that it's actually quite thick. It's way too thick. And you know what? I don't care so much. I'm just going to leave it as it is. I'm just going to leave it as it is, and then I'm going to check again next week how it looks like. However, I would like to now have a quick look right now as well. Let's see how this looks like under the microscope. And that's the one from before. Okay, that not nice looking. Look at this. The cross sections, it looks pretty horrible. Here are some leaves even. This is not good looking. I was actually hoping for much nicer cross sections, but you see they're way too thick. Way too thick. Lots of bubbles. Okay. I am not really happy with the results here. Not really happy with the results here. Okay. But let's see. Yeah, it's also a lot of sections for one slide. It would have been probably better to have one nice one, but you know what I'm going to try? I'm going to actually try this one over here, which is currently mounted in water. I'm going to try to get this one off again and put it on the slide. That's this one over here. Let's try this one again. So we are going to make it warmer. Going to grab again a little bit of this jelly on here. Yeah, it's still warm enough. It will actually slowly start to melt. Let's turn it on again a little bit. Turn it off. So somehow I need to get this thing off here. Let's carefully lift it off. Okay, it's still here. Good. Yeah, let's wait until this actually starts to melt. It probably needs to be a little bit warmer. Okay. Oh yeah, now it's going. Here we go. Here we go. It's melting, 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 melting. Again, not too hot. We don't want to damage it, because if you overheat gelatin, then it will not solidify anymore. So, and now the challenge, am I able to transfer this small specimen over here, this small cross section over here? Am I able to transfer this over into the glycerin gelatin without doing too much damage to it? Maybe I'm going to try the following. I'm going to dip it in here. Pick it up. Pick it up. Ah! Did this work? Or is it still? Ah, it's still here. Okay. It is still here. It's still here. Okay. I think now I might have gotten it. This is really a stressful thing. It's not in here anymore. It's in here. Good. I can see it. Okay. Good. I can barely see it. It's in here. There are still a couple of bubbles on here. Maybe I'm able to remove them by heating it up yet more a little bit. And let's give it a try. This one is wet. I'm going to dry it. By heating it up again, you can again, yes, you can undo the mount, unless the slide is ringed. Some of the and as a matter of fact, I've also read and heard that people have actually done this deliberately to make to fix a slide. Yeah? where the mount has become bad. Yeah, but when the slide has been ringed, then this is of course not so easily possible. So now that's actually one of those cases. If you don't drop it right in the center, then yeah, it's not moving very much good. Yeah, so you can see that it's a fairly thin layer, and over here, this is where it is. Yeah, so you see that over here it's still air. Yeah, it's proof of concept. I'm just going to leave it as it is. Let's have a look. Here we go. Ah, look, it looks way brighter. It looks way brighter. Yeah. Yeah, I'm reading the comments here. I've reheated a few slides to fix bubbles on old antique ones, but those were resins rather than glycerin. I see no reason why it wouldn't work though, as long as the ring is not damaged. Yeah, so I've read some folks actually have completely removed the specimen. They have reheated it, completely removed it. Sometimes even put it into some solvent to remove and soften the mounting medium. Yeah, basically everything's possible. The question, of course, is always, if it's worth it. And with some rare slides, it might well be worth it. So let's have a look here. Here and here we see also the chloroplasts of the stem. So that's actually quite nice. Yeah. So yeah. And if I were to use, now you're gonna see why glycerin gelatin, because if I were to use any solvent-based mounting medium, for example, the one the Uporol, which I commonly use, or there are many others, then this would be a problem because it would dissolve the chlorophyll, the green color, and it might also make the cells shrink because those solvents will also pull out water. Right, so this is a problem. So for this reason, for delicate water organisms, water plants, they always recommend glycerin gelatin. Yeah. 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 So there's a communication going on now by kind of removing the ring of a microscope slide. Some microscope slides have this beautiful round ring around it to prevent moisture from entering the specimen. Yeah. Yeah. This over here. This is an example of where, yeah, it was cut diagonally. Yeah. So another thing that I want to try now, people, is because we're already making slides. I decided now to make also not one of a cross section of a stem, but actually of a leaf. I just want to do a whole mount of a leaf of one of these things, and also using glycerin gelatin. Yeah, for your info, if I were to actually make I'm just doing this for a trial, right? If I really want to make some nice microscope slides, then what I usually do is I use a round cover glass. I usually really try to center it properly and everything. I'm not doing all of that because I want to simply, do a little experiment to just see if this actually works. Okay. So again, a little bit of this gel. Gel is not glycerin, gelatin jelly. Yeah, and again, this one, the one that I'm using here, is actually intended for pollen. It says here for pollen. Yeah, you don't see it. Yeah, no, you can't see it. I'll show you later. It says for pollen, but they also have another one which does not contain the red stain, which is a more general purpose. It's again pretty warm here, so I'm going to lift the. You don't see this. Yeah. I'm going to lift this again here. Yeah, let's put it on here. Allow it to melt. And yeah, let's see how this works. A little bit like this. So there is no direct contact now because it's now on the rim here. So, and let's see, let's take a leaf. Oh, there's some leaves here. Let's take this one here, for example. And of course, we have to make sure that there's all a little bit of mounting medium on top as well. So this one I'm just not going to use because it's still a little bit solid. Put it here on the side. And a cover glass. These cover glasses are not so clean. The reason is because I washed them, but I did not do a very good job. So, and is it big enough? Yeah, let's carefully drop it on here. Yep, it looks okay. This looks a little bit better even. Yeah, and let's see. It's okay. Let's see how good this looks under the microscope. Here we go. Yeah. The chloroplasts are not quite as well visible as in a fresh sample. But it's okay. It's also a question, a little bit of refractive index, and the whole thing has been also heated a little bit. So I'm just reading a little bit. Why don't you put glycerin jelly on the slide with cross sections? You mean the cross sections of the stem? I have actually done this over here in the previous one. Let's have a look here. Okay, so that is now Oh, okay, it's a little bit too bright. Yep. In comparison to the fresh leaf, of course, this one does lack a little bit of contrast. But the cell walls are visible much better here. So I'm going to now. There seem to be bigger cells on top of smaller cells. Yes, this basically, if you focus through the leaf, you can actually see that there are two different layers of cells. Okay, as different ones go in focus, others go out of focus, and then you can actually see that this specimen, this leaf, seems to have two layers of cells. Yeah. Last time you transfer cross sections to glycerin jelly. Yes, I've done that. The one that I transferred before is over here. Where is it? That's the one that I transferred just before. Yeah, that one is now also it's a cross section. Yeah, that is now also in glycerin jelly. Yeah. I'm just reading the comments again. Yeah, yeah. You're talking about shriveling, right? That there's a problem when actually specimens start to shrivel up because when they're in contact with alcohol, that is indeed an issue as well. Generally, I think plant cells might be a little bit more, actually, yeah, more stable because they have a cell wall, but they too will start to shrivel up if you, if they lose water. And this can cause artifacts. This basically means the cells and the structures they don't look like in the natural shape anymore. Yeah, yeah. The transfer was not necessary. Well, the reason why I transferred it into glycerin gelatin is because I wanted it to be a permanently mounted slide. If I keep it in water, then of course the water is going to evaporate. But in glycerin gelatin, it will hopefully be preserved for much longer. This is the whole point of using a mounting medium with all of the advantages and disadvantages, of course, because when it's mounted in a mounting medium like I've done right now, it's preserved, but of course, the process of preserving it might also change the shape of the cells. Yeah? So that's a little bit the thing. And with some mounting media, of course, as we've seen before, it might actually damage the cells. Yeah? So that's again also the leaf, yeah, as such, mounted in glycerin gelatin. So for me, that's the first time that I'm actually making a permanent mount of this specific water plant. Yeah. Why not put glycerin gelatin into the cross sections without transferring the cross sections? I'm a little bit confused. I don't fully understand. How do you want me to put the glycerin gelatin into the cross sections without transferring them? Well, I have a slight understanding problem here. What I have done is I've transferred. The difficulty that I had was before is I had a very nice cross section. Maybe that's what you mean. I had a very nice cross section in water, which looked nice because it was nice and even. And I transferred this cross section then into glycerin gelatin because it looked nice. Yeah. Of course, I could have also made a fresh one, but I just wanted to reuse the one that already worked well. Yeah. Any benefits to wicking out gel from the edges to decrease the thickness? What is possible? I mean, that is actually also done. I just showed this to you as well. With yeah. Will it live long enough to still produce air bubbles? Probably not. Probably not. It's a good question, and the reason why I don't think is the following, because it was slightly warmed, right? Not very hot, but it was slightly warmed. This might have actually damaged the cells. And number two, they've added a disinfectant to this, right? Actually to keep it free of bacteria and fungi so that it doesn't turn moldy. And it smells like phenol, like a disinfectant. So I'm quite sure that this substance will also be damaging to the cells. And plus, there is also the stain fuchsine. It's called fuchsine. That's the pink stain was also added here. So all of these substances, I'm quite sure somehow will damage the cells. That they will probably stop doing photosynthesis fairly soon. My guess. Yeah. I also don't know because if the gel, because it turns solid, might not also itself inhibit the formation of bubbles. Yeah, so here's a bubble. I mean, we can try to check later if it actually started to grow, but I probably doubt it. But I'd like to be convinced of the of something different. But we can check later if it actually started to grow. Yeah, these are oxygen bubbles. Yeah, yeah. I just had a slide with cross sections because they looked so ugly, and now they're completely dry. Okay. So what I, however, could try to do is the following. Because I'm going to simply do now the following. I'm going to take a thicker stem. Here it is. I'm going to take a thicker stem. Just going to rip it off here by let's put this on the side. This is how it looks like now. I don't know if should I take off the leaves? Actually, I should. Yeah, because they might actually disturb a little bit. This stem is actually quite a bit thicker. So maybe it's going to be easier. Maybe it's going to be easier to cut. Look at this. I need tissue paper. And then, yeah, next week we're going to look at it again and see how it looks like. So I think this is actually probably have the instinct. My instinct tells me that this might actually work a little bit better because it's a little bit thicker. So let's put it in here. I see this is down here is too thick. Let's try that again. So all of those leaves here. So here we go. Let me push it down a little bit. So here we go. Yes, this really cuts nicely. 1, 2, 3, 4, 5, 6, 7. See if this is enough. It was not enough. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Yes, see, this was actually quite a nice one here. This tiny one here. Should we, I don't know, I'm gonna just save it quickly on here on this slide. Quickly save it here on this slide. Maybe with a tiny amount of here we go. I don't know if you're able to see it, but it looks almost so my belly tells me that this might be actually a better one. Here's another one. This was not a complete cut. Number 2. Number 3. This seems to be slightly thicker again. Yeah, of course, the danger is that it's starting to dry out now. How am I gonna get it off? Sticky. So I'm going. You're not even able to see this, what I'm doing here. Yeah, it doesn't want to get off. Here we go. Okay, let's give it another try. I'm going to now try to melt the glycerin jelly directly on here. Okay, and let's see if this actually works. And I'm going to, just for the fun of it, try something different. I don't know. Should I drop the cover glass directly on it or a little bit later? So of course, here we go. So, and let's try to melt it again. In the meantime, I'm checking the comments. Ah, yeah, that's the one. Yeah. So, and I'm basically now trying to melt it directly on here. I don't know, maybe, I don't know if it's a good idea to put the cover glass directly on it already before. No, I'll just think. Okay, it's starting to melt. Starting to melt again. Again, this is one of the more difficult to use mounting media. So, and carefully again a cover glass. Also seems to be slightly on the thicker side, some of them. So let's give it a try in any case. So I will save those slides for next week. So that's too thick, I would say. What about the other one over here? Let's go down a little bit with it. Ooh, a lot of bubbles, right? A lot of bubbles. Let's go yet further down with the magnification so we get a better overview. So that is one of them. That's another one of them. and that's the third. That's the thinner one. So, and I think we can already conclude one thing, and it's also something that I learned now. Maybe it is not a good idea to actually heat up the specimen together with the glycerin gelatin, because look at all of those bubbles here. Huh. Yeah, so it would be that would be the next experiment, to basically heat it up, together with the mounting medium, or separately. So over here is the next one here. This was so those cross sections are probably also not quite as nice as I hoped them to be, but that's not so important. I'm more interested now in the sequence of steps, because the next thing that I have to probably do is I have to probably degas everything. Degassing means I have to put maybe the specimen into a vacuum chamber to pull out all of the air. Yeah. Ah, yes, that's the thing. The vacuum degassed the jelly. I think that those bubbles are actually not directly from the jelly itself, but more from the spaces, from the air spaces in the in the specimen. Also, maybe because they were a little bit dry already. Yeah, because of course, as the water evaporates, it starts to pull in air, and they start to shrink a little bit. That's what we see over here as well, and this might actually be the reason for those bubbles. So I think what we should try to do is we should try to keep it actually wet all the time, to even prevent air from entering those cells. Okay, so that is something. Yeah, so we're developing a little bit the method here. Yeah. So yeah, where are these? I lost this here. Here it is. Here's another one. Where is it? Here. Here it is. Yeah. In any case, interesting nevertheless. Yeah. So what else do we have? Yeah. What time is it? What I'm gonna do is I'm going to rapidly switch topics now. Okay. We're gonna revisit this next week again. So we're gonna have a look on how well this actually started to how stable this mount actually is. So I've made now, just in summary, I've made now 3 different mounts here. Just going to show them to you again. This is the one with leaf, right? Then the one that I made right now, and this one is the one which I had originally in water, and then I transferred it directly over into the glycerin gelatin. Yeah, so all 3 of them basically are now different. Same plant, but with slightly different ways of preparing it. Yeah, so I will stop with the water plant now, and now I'm going to make a radical change in topics. Now I have to find a space where I'm going to put those. I'm going to label them later, obviously. I would like to now do the following. I now need a, so this over here. I'm going to use this here. Yeah. So, and I will now try something with this. This here is the water sample from last week. And remember the one from the beginning of this live stream where I basically took a small sample here? This is a concentrated algae sample, directly the same one from over here, only concentrated with a centrifuge. And what I would like to do now is the following: is I would like to enrich some ciliates which might be in here. We're going to now, first of all, have a direct look of what actually is in here. So there is some stuff floating here on the surface. Over here. And I don't know if this is enough, really. Yeah, I'm going to just show this to you, and then cover glass goes on top. Yeah, and then let's make an enrichment culture, and then I would say I'm going to call it a day, because I've already we see. So this is basically the fresh sample from here. So this is how it looks like, and what we're able to see here are, of course, a whole bunch of ciliates, lots, or not many, but some rotifers and so on, right? So this one here, the large one, is a rotifer. You have a whole bunch of single-celled organisms. We've seen this already last week. Yeah. But what I would like to do now is I would like to actually enrich those ciliates yet more. And and I will do that using a wheat grain. Okay, so what I need to do therefore is no idea what this is. Looks like actually like textile fibers. Yeah. But you get the idea, especially at the surface of the water, where the oxygen is yet higher. This is also where you see those yeah, tiny microbes over here. Yeah. So how do I do that? I don't have a fluorescence microscope. I'm just reading this. Yeah. So unfortunately, but you have to draw a line somewhere. There is no limit in microscopy. You can, yeah, there's no limit. Yeah. So I'm going to throw this here as also away. I'm going to wipe this off a little bit. So, and what I'm going to do now is the following. There are a variety of ways of how you can, how you are able to feed them, but I always use those wheat grains. And what I now need to do is the following. I take one of those wheat grains and I somehow, yeah, of course it contains starch. There's still the seed coat on it. So this means there are also, of course, other nutrients present. But one of the reasons why this is so much of an advantage is because it's fairly compact and it doesn't fall apart quite as much as it could. So, for example, of course, people have tried rice grains as well, but the problem is especially with polished rice, you put a rice grain in there and it starts to fall apart. I want to have this actually together a lot. And this next thing is I do have to crush it a little bit to expose the starch. Okay, this is it's very hard to do. Yeah. Maybe it's a little too dry already. You normally use two spoons to crush it. Okay. This has already turned quite hard. Okay, so what I'm going to do now, do I have another one, a slightly stronger one? If not, I'm just going to drop it in as it is. The microbes are going to find the food and then, here we go. This was better. Yes. You see those things here? Yeah, maybe not the small ones, but the larger pieces here, they contain starch. Yeah, they contain the smaller ones, maybe not. And later on, it's easier for me to use tweezers to pull it out again. Yeah, so I'll be using those larger pieces, and I'm going to now do the following. I'm going to I'm just going to drop it in here, and yeah, they're going to sink to the ground. And I'm going to use another one over here as well. This is, and I'm also going to crush this here. Here we go. Yeah. Starch is exposed, but the seed coat will hold it together. Yeah, so it's easier to take out again. Blup and blup. And that's it. And now I have to wait for a couple of days. And then I hope that I'm going to see hopefully more less interesting microorganisms in this crushed wheat grain. The reason being is that, of course, bacteria start to multiply. Yeah. But at the same time, those bacteria are also eaten by the ciliates and rotifers that are in there. If you now were to add milk, it is called the so-called a milk culture. Then a tiny amount of milk. Sometimes this also works, but there's a real danger of adding too much milk. Then everything might go out of balance and starts to decay and smell bad because the bacteria grow far too fast compared to the other larger ciliates which are able to feed on them, right? And by using a wheat grain, what you're doing is you're actually keeping keeping the microorganisms concentrated around those wheat grains, and the rest of the liquid is not quite overfed as much. Yeah. So this is the thing here. Yeah. Yeah. There's some recommendation concerning safety, eosin stains. Yeah. So of course you always have to be careful with with chemicals. Yeah. Yeah. Do you know what I've also tried, and maybe maybe CT Lab because you're doing just a question to CT Lab, one of the people who comments here. What I've done is a long time ago, I've been actually experimenting with fluorescent markers. In German, you call them Leuchtstifte, highlighters. The yellow one, and they also start to glow in UV light. And I wonder if you can use this as a stain. Yeah, so I remember a long time ago, 30 years ago at the university, we had a fluorescent microscope. We played around with that a little bit. I tried to squeeze out some of this fluorescent ink from those highlighters, from the Leuchtstiften, as they're called in German. And it was actually also, it started to glow. That would be kind of interesting whether they would actually also react with some parts of a cell to be used as a fluorescent stain. Yeah, or here's the answer already. Yes, it's possible. That was fast. Thank you for the response. Yeah, this would be interesting as well. But again, in fluorescent microscopy or in any kind of staining, of course, it depends with what the substances actually react with. Yeah. Yeah, so that's basically the thing. So I'm going to slowly call it a day again for today. I'm going to now leave this here in this jar for next week. Okay, then we're going to directly observe what's in there, if there's anything in there. There will be, but I hope something interesting as well. And then we're going to also revisit those 3 slides that I made today to see how good they actually are in preserving this. I know that some of the, how do you say, preparations were a little bit very fast, but when I have a little bit more time now over the summer holidays, I might spend a little bit more time in making a little bit the nicer cross sections. I'm going to call it a day today. I'm already a little bit tired and my voice is starting to give out as well. Yeah, I'm going to add here a little bit more water to make sure that these algae continue to grow as well. Yeah, I hope that also my other Eludia water plant is slowly starting to regenerate. Yeah, in any case, I wish you all the best. Yeah, and yeah, hope to see you again. Bye bye. Happy microbe hunting as always, and see you around. Bye bye.